Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and very good morning or afternoon. Welcome back to SID2004. We're going to continue our lecture. I'm a bit tired today, even though it's just the start of the day. Why? Because I've already actually completed a recording of this lecture. However, no audio was recorded. Therefore, I need to re-record everything. I need to clear up all the notes or scribblings that I did and record the lecture again. Okay. So, but anyhow, that's my job. I still need to do it. So as I mentioned previously, when I have all these recorded lectures posted on Spectrum, if you actually watch it on Spectrum, there will be some tutorials. There will be some inline questionnaires that I've put in. So if you do like that kind of system, do help me by going to this link. Okay, do a QR scan on this link and tell me whether you like it or not. By no means, please just do not compare this with a face-to-face. What I'm looking at is that do you like a live lecture or a synchronous lecture with all these questionnaires embedded as a practice while you are listening to the lecture. So those are the things that I'm looking at. This is by no means a feedback to the university saying that I want to come back to university. Okay, so this is not it. This is just for me trying to see what would be the best way for me to teach you guys so that you can get the best out of it. Okay, all right. So last week we've looked at just a short bit of chemical synthesis of proteins and peptides. What we will do today is well, this week, so to say, okay. So is to look at again in peptides and proteins in a little bit more detail than last week. So if you look at the schedule that lecture timetable that was given to you earlier in the semester or earlier during the lecture on week 8, you see that underneath over here it should be a tutorial to discuss about this quiz. But it's very unfortunate that we have two public holidays during our lecture slots. Therefore, I need to do a little bit of a catch-up session with the contents of lectures, and of course, I need to sacrifice something. So I decided to sacrifice is instead of sacrificing the quiz, I cannot do that. I still need to have a quiz, two quizzes. So we've done one 2 weeks ago, last week, and we will have another one the 2 weeks in 2 weeks time. So what I will do is instead of having a live tutorial discussion, I will do hopefully I do have time to actually do a recorded lecture on the or a discussion uh, my own by just myself, recorded answers of the quiz number 2. Okay, I will post the quiz on Spectrum, and it's up to you guys if you guys want to have a look at the answers. Then by all means watch the videos. Otherwise, you don't have to. Okay. All right. So I'll just skip through all the non-essential things and move forward, because as you see here, I only have 2 more slots for to talk about peptides and proteins, and next week we will delve into nucleic acids. Okay, so my original plan is for you guys to view nucleic acid first and then relate back to peptides and proteins. But since, well, according to the plan, we have a lot of public holidays, I'm just afraid that we'll not have time to actually cover everything about peptides and proteins. So that's why I do need to take a slot for peptide and proteins. And if I manage to cover both amino acids in next week, then we will have a tutorial over here. Okay. Again, this is just a planning. I'm hoping for the best. Okay, so what we looked at last time was the strategies to options to actually synthesize peptides. So if you just mix two amino acids together, it will not spontaneously form a dipeptide. So you need to have a way by which you can control the reaction, either to promote it or to inhibit it. Okay, so and the first strategy that we've looked at is the protecting group, the use of protecting group. Okay, so of course, in an amino acid, you have the carboxyl terminal, you have the amine terminal, and sometimes the R groups you do have a functional groups that are either a carboxyl or an amine as well. Okay, but for this sake, we'll just look at in very general. We just look at either the C terminal and the N terminal and how you actually grow the peptide from the N to C instead of looking at the side groups, okay, side chains. Okay, so P is abbreviated as the protecting group over here, just to make sure that you guys recall. Now, you have two ways of doing this. One is either you protect the protecting group at the N terminal, okay, so you protect at the N terminal and then you do a coupling. On the right hand side is the protecting group on the C terminal. In the middle, protecting group is an N terminal, okay. If you have this type of peptides whereby you have all functional groups protected, what are your options? So your options is the first one, the most right, is to remove the C protecting group and then you couple the next amino acid on the C terminal. So what it means is that you are basically growing the peptide from N to the C terminal. Okay, so that's the first option. The middle option over here is by you remove the protecting group at the N terminal. So now you have the first amino acid, you have the C over here. So you remove the protecting group at the N terminal, and then you add in additional amino acid. Thus, you can grow it from C to N. Okay, so these are the two options that you have. Of course, if you just remove all protecting groups, you cannot grow them all together, but you will get your dipeptide of interest. Okay, so these two options on the most right hand side and the middle one are ways by which you can grow not just a dipeptide but a tripeptide and so on and so forth. Okay, so we will look at all of these in a little bit more detail and what are the options that or reactions that you can actually consider. Okay, knowing protecting group is one thing, but the most important thing that you need to know is what are the functional groups available at the C and N terminals, and what are the chemistries that you want to use to protect both C and N terminal. Now, there are so many reactions that you can do in both C and N. However, because what you are doing now is look at this slide again, what you want to do is you want to grow the peptide. Okay, so you want to add in more amino acids after another. Therefore, you need to choose the best protecting group that is easy enough for you to remove and add. At the same time, you need to make sure that the protecting group of your choice is compatible to one another. Okay, so we will look at compatibility towards the end of the lecture, but for now, we will just focus on two functional groups. You have the carboxyl highlighted in red, and you have your amine group highlighted in blue. Okay, so the best strategy to do this is by using esterification at the carboxyl terminal and acylation at the N terminal. Okay, so you've learned about esterification during your first year. You've learned about acylation is also during your first year. So you know that both of them are the most basic chemistry. Therefore, if you can use it simply by just adding and extracting protecting group, then that's the best choice. And of course, this is what researchers have been doing, looking into simple chemistries to promote addition or removal of protecting group. So amino acid reacts with alcohol in the presence of acidic catalyst. Simple enough. That's the basic reaction of esterification. So under acidic condition, the amino group protonates, thus will not interfere with esterification reaction. Okay, so you originally have your carboxyl and amine uncharged originally from the previous slide. You do esterification, you protonate the amino group. Therefore, when you add in your alcohol of interest, the protecting group, so to say, you will just couple on the carboxyl terminal, forming the ester. Okay, as simple as that. H3 plus. Oh, 3 is over there. Okay, so now you can add in the ester, and of course acidification. If you want to remove it, you just go back to doing more acidic or hydrolysis or a basic hydrolysis. Then you can simply remove it, forming again your carboxyl terminal. Okay, so as I just mentioned, okay, so you can regenerate amino acid by simply doing a hydrolysis process. Acidification is used to protect carboxyl terminal, removing its acidic characteristic. This is to prevent the carboxyl terminal or carboxyl group from reaction in some undesirable manner. Okay, undesirable manner meaning that we will look in the next few slides, by which, as I mentioned, if you have two amino acids A and B, if you react them together, what are the possible outcome? You can get AB, okay, you can get BA, you can get BB, and you can get AA. And of course, if they are not protected, then you can have multiples of these, 3 copies, 4 copies, and so on and so forth. So there are unlimited copies. So if you protect the carboxyl terminal, at least you will stop some reactions from happening. So if you protect B at the C terminal, for example, okay, this one will happen. Okay, you know that one will not happen because you now have a protecting group on the C terminal. Okay, so you will not get a BA, and of course you will not get BB. Okay, you will still get AA, but you will not get multiple copies of B. Okay, so now you are eliminating at least half of the possible outcomes. Okay, so common protecting groups of esters, PG of esters that you can use is simply like methanol, ethanol, T-butyl, and so on and so forth. So any alcohol, you just name it, you can just simply use it, basic ones. However, due to high probability of racemization from the C-terminal synthesis, this approach is hardly used nowadays. Okay, so what do I mean by that is that if you look at the structure, you do have the chiral center over there. Okay, so this proton on the chiral center is what we call as acidic proton. So under the presence of base, for example, if you do hydrolyze it using a base, you can deprotonate that one as well, and therefore creating a racemization. Okay, N2C approach is not feasible. So N2C is growing through the C terminal, and therefore it's better to use the C2N approach. Okay, but nonetheless, N2C approach is being used widely in the nature, okay, in our body, for example. So looking at the second options where we have the amine group, how do you actually do it? Acylation, which is the C2N approach that I just mentioned. Okay, it's the most common strategy for protecting the amino group and reducing its nucleophilicity is to react amino acid with carbonyl derivatives. Okay, the easiest way to do it. So for example, over here you have the CBZ group, an acid chloride, or you have a Bock anhydride structure. Okay, you do have a delta positive on the carbonyl carbon, while you have a delta negative on the oxygen. Okay, for both. So if you have an amine group with a lone pair, you can easily react it to there, you protonate this, and you can get an amide bond protection very easily. Okay, an acylating agent, for example the Bock anhydride, the figures that I've shown you just now, converts the amino group to an amide, as I just mentioned. The reaction most commonly used for this purpose is again same thing. Amino acid and Bock anhydride will give you our protecting group, which is known as Bock group. In peptide chemistry, we call it as Bock. So what are the chemistries involved? As I mentioned, you have a lone pair of your amino group attacks the delta positive under catalytic or basic conditions. Then you have either capture proton and then return it back and then cleave off this um, um, anhydride bond. Therefore, you will have your amide bond. Okay, that one is C terminal OH and your R group. Okay, as simple as that. But of course, you will also might get some of this going on over there, but the amino group is more reactive, it's more nucleophilic. Okay, doesn't mean that the other one doesn't work. It does, but again, to a certain degree, the amino group is preferential in this particular reaction. Okay, because Bock is an acid labile or base resistant. Okay, so we need to remember this terminology. So acid labile means it's base resistant. Okay, acid labile meaning that it's will react under acidic condition. So it will not react in base. So we call it acid labile base resistant. It can be removed using a strong acid. So again, you have a bulk, you have a strong acid, for example, trifluoroacetic acid in this case,
Then you get the deprotection.
of your amino acid. Okay, and at the same time, you have effervescence of carbon dioxide and your byproduct. So now we've looked at the first strategy, which is protecting group,
So what is the second strategy?
is by using an activating agent. Okay, as I mentioned, the amino acid or two amino acid does not spontaneously form an amide bond. So if you put it together, it does not spontaneously form an amino acid. It might take a few years. Yes, you might get a dipeptide, but then again, the reaction is too slow because the carboxyl terminal is unreactive. So you need to add in something else to promote the activity of the carboxyl terminal, and that is what we call as the activating agent. Okay, so many types of activators are available. We looked at over here is what I call as DCC, or NN dicyclohexyl carbodiimide. Okay, um, what is the structure? The structure of DCC is like this. So you have a dicyclo, NN, because it's connected to an N dicyclo, and you have the carbodiimide in the middle there. So DCC is one of the early generation of carboxyl activator. There are some other ones that are called as DIC, EDC, and so on and so forth, but these are all first generation activating agent. They are still widely used, not just for peptide chemistry, but any reactions that aims to promote formation of an amide bond. Okay, again, if you recall, you want to produce an amide bond by coupling of two amino acids. So similarly, if you want to form any amide bonds by coupling, say, lipids with um, whatever amine, you can still do it using DCC, DIC, and so on and so forth. But for peptide chemistry, this is the first generation. The second generation, third generation, and so on and so forth, is because DCC still promotes certain degrees of racemization. Therefore, the second generation and third generation of carboxyl activators tend to try and reduce the well, you want to activate the carboxyl terminal, at the same time you want to reduce the possibility of racemization. Okay, so DCC activates the carbonyl group by putting a good leaving group at the carbonyl terminal. So that is the reaction scheme. So you have your amino acid or just simply the carboxyl functional groups. So as shown here, you have your DCC. DCC promotes activation. Okay, you form a stable intermediate. Okay, so this is important. And then from the stable intermediate, stable enough that you can isolate it, relatively stable. Nonetheless, it activates the now carbonyl. Okay, so in the presence of a second amino acid, you can simply add in and manage to get two amino acid coupled to one another. Okay, and of course you will have your urea as the byproduct. And as if all reaction, you want to get rid of the urea before you actually continue with your synthesis. Okay, so although this lowers the energy for an amide bond to form, the reaction by no means free from other side reactions. Okay, so just now I've shown you guys about this side of this image, but additionally what you will get by using the first generation of activating agent is you can get a formation of reacting of the activated species. So that one is the activated species. You can get the same amino acid, see R1 over there, coupling to form an anhydride structure, and then you can get you can still get the same amino acid over there, okay, the same dipeptide, but additionally you are wasting resources because you are now doing an additional step. So that one is just 1, 2, and 3. But now if you were to go into a different path, it will become 1, 2, 3, 4 steps. Okay, so in terms of synthesis, this is a wastage because you want your reaction to turn from a reactant to a product using the minimum steps as possible. Okay, if you want to relate it to energy level, remember when you have energy level like this. Okay, so you want your energy level to be your product between your reactants and your product to have a minimal level. Okay, so regardless of the pathway, then the energy level is always similar. But for a 4 level reactions, you might have 4 steps, 3, 4 before you go down. But for 3 reactions, you only have 1, 2, 3, for example. So you are saving this much of energy over here. Okay. So therefore, to minimize the other side reactions, co-activators or reagents are normally added to promote the formation of two and minimize the formation of these unwanted reactions. Okay. For example, you can add in a base. Okay. You add in a base so that it will deprotonate your amino acid at the C terminal. So you want this to happen to a certain degree, but at a certain degree you don't want a side reaction to happen. So if you manage to deprotonate that one after it forms this stable species, therefore you can reduce this side reaction from happening. Okay. Also, you can also use, for example, N-hydroxyniamide to just kind of like helps in reducing the side reactions. Okay, so basically that is the general principle. So 2 strategies for a liquid phase, whether you want to do it N to C synthesis or C to N synthesis, both are fine as long as you are using these 2 types of strategies. Okay, but there are still one major drawback if you were to synthesize peptide using a liquid phase. The major drawback is that you have your amino acid, you have a base, now you have your reagents. What you will get towards the end is multiple mixture. What do I mean by multiple mixture is you might get your dipeptide, okay, you might still have your base, you will have, if you were to if you want to deprotect your protecting group, then you will get your protecting group in the mixture. You might get some byproduct of the activating agent, for example the urea byproduct that I've shown previously. Okay, so it's fine if you have this because you can always purify them, right? Okay, but purification process is lengthy. Okay, so that one is one thing, so you can purify them, there's no issue. But if you were to not purify them, because purification takes a while, and then for example, if you were to do a column chromatography, how long will it take you? It will take you so much time to actually do a column chromatography. So if you want to skip the purification process, and then how do you proceed with the third amino acid coupling now that you have a lot of other byproducts in there? So you can do chromatography, of course. Okay, lots and lots of chromatography. You can purify them, then you can get your peptides. No issue. And then once you get your dipeptide, a pure one, so you can just repeat the process, add in your third amino acid, okay, third amino acid, and then followed by chromatography, and then you get a pure ones. You do a fourth um, addition of your amino acid. You do it back again, and then go back and forth, and so on and so forth. But chromatography takes a lot of time, therefore alternative mean is very, very necessary. So how can you do it? So there are 2 approaches by which this option or this peptide synthesis can be simplified. So the first option is still in solution phase peptide synthesis, okay, by using a reagent that produces volatile byproduct. Okay, so this is the keyword, volatile byproduct that can easily be removed during chain growing. So it's still a conventional, it's still as what I've shown you previously, but if you have a volatile byproduct, instead of doing a column chromatography, which takes you a few hours to pack the column to make sure that there's no air bubbles in the column and so on and so forth, which will take forever, you can just simply do a rotary evaporator. Okay, so rotary evaporation, you manage to remove the volatile product, then you can just repeat the steps. So rotary evaporator is well, it doesn't require as much time as if you are doing a column chromatography, but nonetheless, if your reaction is say 2 or 5 liter reaction, then you still need to take a lot of time to actually remove the volatile byproducts. Okay, so the second alternative, the second approach is to use a solid phase peptide synthesis. What do I mean by that is by, as the name mentioned, you use a solid support to grow the peptide. So we looked at these two examples in a little bit more detail. Okay, so first thing first, let's go back to the basic. Say if you want to synthesize a tripeptide like this, glycine, lysine, alanine, and glycine. Okay, how can you do it? Of course, we have two options, N to C or C to N, but because N to C has issues, so C to N is more common. Okay, so you can use both C to N for both SPPS and SPS. Okay, so C to N meaning again you have the C terminal as the first amino acid, and then you grow it towards the N terminal. Okay, second amino acid, third, and fourth. So this is how what modern peptide synthesis does. Always goes from C to N. Okay. So SPS works well for small peptides, but many peptides and many peptides have been synthesized in this process. So if you go back and look for the history of peptide synthesis, you will notice that the one of the earliest peptides that are being synthesized, which is the insulin, was synthesized from C to N in the solution phase. Okay? It takes so many years, not months, years to actually complete this. Okay? A long time ago, but nowadays, you can easily synthesize insulin within a week. Okay, or if you have a better system, you can actually synthesize it within 2 days. Alright, a large number of chemical reactions and purifications are required, even for a small peptide. As I mentioned, purifications here meaning a rotary evaporator or column. Okay, small peptides. Well, if you have only 2 peptides, you need to have 1 purification. If you have 3 amino acids, then you need to have 2. The longer the peptide, the more purifications that you need to have for a solution phase. So although the individual yield are excellent, with large peptides, the overall yield becomes so small to be unusable, so unfeasible. So the synthesis is no longer feasible. And it might take years or months to acquire a complete synthesis of a whole large protein or peptides. Okay, so a large amount of times require an overall yield largely due to lag in purification. Okay, so even though SPS works well for a very short peptide, but there are still disadvantages. Okay, let's look at the disadvantages in a little bit more detail. So say if you want to still synthesize instead of doing a 4 amino acid as I shown previously, say if you want to synthesize 20 amino acids. Okay, and in practice, okay, when you learn about chemistry, there's always a yield involved. Okay, and you've been doing reactions in the lab previously, you know that there's no reaction that produces 100% yield. Okay, so that's why I said in practice a reaction never goes to 100% completion. Even if you get 80% completion, okay, it's already good enough. So imagine if you want to synthesize 20 amino acid, and let's say each reaction has 80% efficiency. So how do you calculate the yield? So by the end of the 20th amino acid, the maximum yield that you can get is 1.4%. So how do I get about to this number? Imagine your first amino acid, okay, 80%. So you get the dipeptide is at 80% yield. So, but once you couple the third amino acid, so the originally 80%, it now becomes another 80% efficiency, you get 64, and then by the fourth amino acid, again 80% efficiency of synthesis, the maximum yield that you can get is 51%, and by the end of the 20th amino acid, the maximum yield that you can get is 1.4%. That is if and only if the efficiency is 80. So what if the efficiency is lower? Then of course the yield that you will get by the end of the 20th amino acid will be way lower. Okay, and on top of that, if you were to use SPS, the peptide must be purified after each step to ensure that byproducts are not there to actually try and involve in the reaction. Okay, if the byproduct is still there, then of course the efficiency will get lower, and therefore the overall yield will be less than whatever is written here. Okay, so the peptide must be purified after each step to prevent subsequent unwanted reaction and with leftover reagents, as I mentioned. So towards the end of the day, the yield will be very, very low, 1.4%. Okay, so take insulin as an example. So originally in 1921, between 1921 to 1969, whereby diabetes was discovered, and people know that insulin can be used to actually control the symptoms of diabetes. So originally it was extracted in pigs and cows. Okay, extracted, of course, purified and injected in humans to be used as an insulin. However, um, increasing of diabetic problems is with allergy to the animal products. So if you were to extract the animals, there are like multiple issues that you need to look at, reactions from allergic, um, and whether the extracted proteins or peptides have been purified properly, without bacteria and whatnot, because towards the end of the day you want to inject it into your body, so you want the insulin to be as sterile as possible. However, insulin, you can simply just heat it up to 100 degrees temperature, hoping to kill all the bacteria. It doesn't work that way. Okay, if you do that, then you pretty much also decompose the insulin, and thus the insulin will no longer work. Okay, so therefore, it fueled research to find an alternative to produce insulin, and one of the means is by doing, of course, because the lecture is about protein and peptide, then there will be protein and peptide synthesis. Okay, so the structure of insulin was solved in 1969 by this researcher Dorothy Mary Crawford Hawkins. Okay, after 35 years using X-ray crystallography, so something that we've learned, um, like it's nothing. Okay, so was originally um, designed and research take so many years to complete. So after the huge contribution of this lady, we managed to identify that insulin has two instead of just one, two peptide chain. So simply by, if you were to go back and see the degrees or the levels of protein structures, then this is, as I mentioned previously, easy quaternary structure. Why? Because it has two peptide chain, and each peptide chain has its own secondary structures. All right, now, so because we already know the sequence of insulin, then we can proceed with total synthesis. However, in 1978, the era of cloning began. So cloning, by which you know about DNA of insulin, the host that you can use, and therefore you can actually produce insulin biologically using a recombinant DNA technology. Okay, so an example is humulin. Humulin is chemically identical to human insulin, but it was produced by means of biotechnology. So the peptide chemistry lags behind because of the discovery of this technology. However, at least on 1963, okay, the solid phase peptide synthesis was born, it was discovered, and thus it actually promotes the field of peptide discovery. So you can still discover peptides using SPPS, but then if the peptide is a peptide of interest, instead of proceeding with a chemical synthesis, you can always go back and do a biological approach to synthesize the peptide. Okay, because one keyword here is it's cheaper than a synthesis. Well, of course, there's a lot of drawbacks and so on and so forth, but we're not gonna delve into that one. Okay, so what is SPPS? So SPPS is known as a solid phase peptide synthesis. Originally, it's known as the Merrifield approach of peptide synthesis. Merrifield is the guy, Bruce Merrifield. You can read his the first paper published about peptide synthesis, synthetic peptide synthesis using a solid support. It's very interesting on how they managed to discover this approach. Okay, and the most important thing is that SPPS has a way bigger advantage than SPS, especially in terms of time it takes to synthesize the peptide. Okay, why? Because SPPS does not need to have a purification step in between the reactions. Okay, intermediates meaning that um, between the amino acid coupling. So again, for a solution phase, after each coupling, you need to wash it. Okay, you need to have a purification steps. The easiest one is rotary evaporator, but still rotary evaporator takes some time. Okay, for SPPS, you do not need to do any purification step. Well, the purification step is very easy as taking a bath. You just wash it, done. Okay, so peptide grows directly on the solid support. Therefore, it's not in the solution, it's on the solid spot. You can easily just take away the solution and you still get the peptide and the solid. Okay, and excess reagent byproducts can easily be removed simply by washing. So what it does is normally you have a peptide vessel. Okay, the shape of the vessel varies. So say for example, if that one is a syringe, okay, at the end of the syringe you can add in a filter. Okay, this filter does not need to be like a very tiny filter, but enough to say that the filter needs to be selective towards a bigger particle. So if you have a solid support up there, okay, so I just draw a syringe. If you have a solid support, so the solid support will just stay in the filter, while if you have a region of byproducts and whatnot, it will just be drained out. Okay, so it's way easier and way faster to drain out a solution rather than to evaporate them. Okay, so that's why SPPS has a bigger advantage compared to SPS. So what does the solid support means? Although it's not 100% solvent free, but the synthesis is linked to a solid support. So this is what it means by a solid support. Similarly, for when you talk about solid phase NMR, it does not mean that it's 100% free reaction. You can still have a little bit of solvents to promote your whatever you are aiming for. Okay, so in this case, you grow the peptide on solid support, therefore you consider them as solid phase synthesis, while the other one, because the reaction is being done in solution, so it's a solution phase synthesis. Okay, and the solid support is actually a polymer beads. A polymer beads made up of polystyrene polymer. So this is the first generation Merrifield resin. We call it Merrifield resin. The original ones is made of polystyrene polymer. But nowadays there are different types of polymer being used to substitute this resin. Okay, but most of the available synthetic resin are made from polystyrene polymer. Okay, so you still do a C to N approach like that. Okay, therefore free amino acid with a free carbonyl carboxylic terminal can be attached to a functional group on the solid support. So for example, if your solid support has a chloride, a good leaving group like that, you can simply react this with the carbon over there to form an ester group. Okay, so this is the first generation of solid support. If I'm not mistaken, there's already like a fourth generation resin, but in my case, I did not use it because it's rather expensive. Okay, so that is all for today's lecture. We will cover just the remaining bit next week. Okay, sorry, next Thursday, and I will have all the tutorials in that particular slide because particular lecture because this lecture is already about 40 minutes, for 43 minutes almost. So yeah, if I were to add in more questions, then you will need to spend more time to actually complete the whole thing. Thus, I will put in um, more questions for the Thursday lecture. Okay, so we'll stop here for now, and I'll see you guys again, or you guys will see me again or hear me next Thursday. Thank you and take care, everybody.